Hi guys, this is David Villar and welcome to this first video on toxicants that affect the liver. I selected key toxicants to discuss among drugs, chemicals, mycotoxins and plants which are more relevant for large and small animals. It's actually impossible to make videos of all of them, but you can consult the list in Beasley's notes if you would like to read more on any particular one that we missed to include here. On this first video, we're going to make a quick review of what to expect when the liver fails or is damaged, and we'll discuss acetaminophen and aflatoxins. Now, the liver is uniquely positioned as the body's primary detoxification center for ingested substances. On this image, we see that blood travels from the digestive tract via the portal vein directly to the liver, where it percolates through the sinusoids with their porous endothelial lining. The hepatocytes are able to take up and process not only a range of nutrients but also many natural and man-made chemicals. What we see in this graph is some of the common biotransformation reactions and elimination pathways. Some substances do not need to be biotransformed and can be excreted intact, but most of them will require phase 1 reactions to be excreted, and even an additional step with phase 2 reactions. Phase 1 reactions include oxidation, reduction, hydrolysis by microsomes, and particularly cytochrome P450 enzymes that catalyze those reactions. Phase 2 reactions are conjugation reactions in which the chemical or the metabolite from phase 1 reactions are bound to a molecule like glucuronide or sulfate that will usually make the excretion with bile or urine easier. All these reactions usually detoxify most foreign chemicals, but sometimes will make them more toxic, as we will see with acetaminophen. We could go on talking about these reactions, but we will refer to them for every specific toxicant. In general, what are the clinical signs expected when the liver fails? The signs in white color are the same whether the animal experiences acute, subacute, or chronic exposure. What are these signs? Well, not very specific. Vomiting, anorexia, diarrhea in most cases, and not always present, but if they do, they are very highly suggestive of liver failure, which is jaundice and ascites, which, as you know, is water belly. That is why I put a plus or minus in front of them. You not always see these two clinical signs in every case. There are other signs that go along with acute liver failure, which is abdominal pain, shock, that is usually hypovolemic if there is extensive hemorrhage within the liver, and occurs in over a relatively short period of time, maybe just 24 to 48 hours. With severe liver failure, the animals can die from hypoglycemia, and upon palpation, the liver can be enlarged and painful. With subacute liver, we can see the same signs as acute signs, but may not be as severe. And with chronic liver failure, there are additional signs that include encephalopathy from ammonia accumulation, hypoproteinemia that may cause fluid accumulation. The animal has probably lost weight and can develop photosensitization, particularly if they're grazing animals. And the liver will tend to be cirrhotic, which is, as you know, hard from fibrosis, and may have actually shrunk and lost up to 80% of the parenchymal tissue. So what are some serum biochemical tests used to detect hepatic disease, also called liver function test, even though very few actually measure a specific liver function? The liver lesions can cause leakage of enzymes that originate within the hepatocytes. ALT, or alanine aminotransferase, is specific for the liver in dogs and cats, but has no value in the horse and cow. AST, or aspartate aminotransferase, is not specific for the liver because it can also originate from muscle. And we have sorbitol dehydrogenase, which is used most commonly in the cow and the horse. So what are the causes for leakage of these enzymes? Well, one of them is lack of oxygen, such as with heart failure or shock. another reason could be a direct action by something toxic, inflammation, metabolic disorders like lipid swelling. If these causes are eliminated, the liver has a great capacity to regenerate, and the lesions can be reversible. The problem is that reversible biochemical lesions cannot be differentiated from irreversible ones. So all we can do is look for the duration of the increase in some of those enzyme values to assess the progress of the hepatic disease. When we talk about cholestasis, the evaluation is accomplished by testing for bilirubin, whether it's conjugated or unconjugated, and alkaline phosphatase. Now the problem with cholestasis is obstruction of the bile flow that may be a consequence of fatty changes, degeneration, inflammation, fibrosis. And when there is chronic damage in which about 80% of the liver may still be viable, 20% of the liver may still be viable, there are other things that can be found commonly. One of them would be albumin, maybe very low because it's not there is not enough production, and this is an indication or evidence of a terminal stage of the liver disease. If you put this together with an elevation of ammonia, it means that there is not enough functional mass of the liver to convert it to urea. And the same thing would happen with biliary acids. They cannot be removed by the liver, just like when you give a dye like sulfobromophthalate, it cannot be removed from blood by the hepatocytes and eliminated with bile. I thought to show you an example of the liver damage that happens because of hypoxia secondary to heart failure. The arrow is showing the direction in which the blood flows going from the portal triad towards the central vein. The hepatocytes close to the portal triad are okay, but as you move away from it, they start to degenerate. They then die and start disintegrating, and then the red cells fill in all the spaces, and you see a hemorrhage. Let's move on to acetaminophen, which is a good analgesic and particularly antipyretic in humans, but is not recommended for dogs and cats. This is a great example of what the liver does to metabolize or biotransform drugs. It is directly conjugated by phase 2 reactions with either glucuronic acid or sulfate, which can be easily excreted with the urine. In this case, you do not want the drug to go through the phase 1 reactions, because the P450 cytochromes, instead of detoxifying acetaminophen, they do the opposite. They convert it to a very reactive metabolite that is very toxic and binds to macromolecules like protein and DNA, and that will kill the cell. The cats lack the specific glucuronil transferase required to conjugate acetaminophen, and at toxic doses, both reactions become saturated very quickly, and more drugs goes through the P450 pathway. Within the cell, there is another phase 2 reaction, which is glutathione that binds that reactive metabolite and converts it to an inert compound that is excreted as a mercapturic acid conjugate. The problem is that glutathione sources are quickly exhausted, and when the levels drop to 20 or 30% of normal, those reactive metabolites will bind proteins and cause cell death. Now this slide shows the molecular structure of glucuronic acid and phosphoadenosine phosphosulfate. The black highlighted part of the molecule is the moiety that will bind to drugs or metabolites and make them more water soluble to be excreted with the urine. At the bottom, we see the glutathione molecule. It's made up of 3 amino acids, glutamic acid, cysteine, and glycine. And it is actually the cysteine in the middle that is the one that will bind drugs and convert them into a mercapturic acid derivative. There is something unique about dogs and cats, which is that they develop methemoglobin even before you see liver damage. In humans and most other species, the toxicity is just associated with liver damage, and you may see abdominal pain, jaundice, vomiting, and shock, as we saw for the causes of acute liver failure. But in dogs and cats, they primarily develop methemoglobin and hemolytic anemia with cyanosis, dyspnea, depression within 2 to 4 hours of the exposure. So how come that dogs and cats develop blood toxicity before liver toxicity? Well, the red cells do not have P450 enzymes, so the same toxic metabolite that is produced in the liver cells cannot take place. There are many chemicals that are known to induce methemoglobin, and they do so by co-oxidation and redox cycling with oxyhemoglobin, as we see on this slide. In co-oxidation, an electron is lost from both the chemical and the iron in the oxyhemoglobin, and that results in the production of a nitroso compound and methemoglobin and a reactive oxygen species such as hydrogen peroxide. So this co-oxidation reaction occurs repeatedly due to redox cycling with repeated reduction of that nitroso-containing metabolite by glutathione, and this is followed by repeated co-oxidation cycles. So this cycle can only be stopped by alternative biotransformation pathways, which is what is deficient actually in dogs and cats. There is a minor metabolite of acetaminophen that will co-oxidize and redox cycle with oxyhemoglobin, and that is paraminophenol. This minor metabolite is also produced by deacetylation of acetaminophen in the liver and is then released to the bloodstream. And when it enters the red cells, most species have N-acetyl transferases that remove the paraminophenol from the redox cycling cascade. But as I say, dogs and cats are deficient in their ability to acetylate in the red cells, and that explains why they develop methemoglobin. So what are the toxic doses in dogs and cats? Well, cats are 3 times more sensitive than dogs at doses of 200 to 200 milligrams per kilogram. The animals develop 20% methemoglobin in 4 hours. All of the animals given that dose recovered without treatment, but they had to go through a period of vomiting, depression, and abdominal pain. When a dose of 500 milligrams was given to 4 dogs, one of them died, and they all developed methemoglobin at 50% in 4 hours, and 3 of them recovered without treatment, but they had to go through a period of severe depression, vomiting, cyanosis, which, as you know, is gums with a bluish color, hemoglobinuria and hematuria, and facial and paw edema, which is also called as moon face. So exactly the same thing happens in cats, but at 3 times lower dosage. The group of dogs with 200 milligrams per kilogram in cats is the same one as with 60 milligrams per kilogram, and the group of dogs on the 5 milligrams per kilogram dosage now becomes the same one in cats at 120 milligrams per kilogram. So as you can see, the clinical signs are the same in dogs and cats, but at 3 times different doses. Here is a picture of a cat with a moon face and edema on those paws. It's not known what mechanisms produces this. It's probably some type of damage to endothelial cells at those sites, but they haven't really come up with an answer for that. So what is the treatment? It relies mostly on replenishing the stores of glutathione and sulfate. The mainstay of the therapy is N-acetylcysteine. You can start with a bolus dose of 140 milligrams per kilogram IV with dextrose, and then follow that orally with 70 milligrams per kilogram for 7 treatments. As an alternative to N-acetylcysteine, you can use sodium sulfate at 50 milligrams per kilogram IV at the same interval and duration. And there is another drug that works similarly to N-acetylcysteine, which is adenosyl methionine, which is also a precursor of glutathione. Empirically, it has been recommended to give ascorbic acid as an antioxidant, and you will find in all textbooks cimetidine to inhibit P450 oxidative pathway, but this is also an empirical treatment. There are no studies to prove efficacy. If there is severe cyanosis, oxygen should be supplied, and actually steroids and antihistamines are contraindicated. So, moving on to aflatoxins, there are a number of mycotoxins that are of concern to animal and human health, and aflatoxins are one of them. They are secondary metabolites produced by some strains of the molds Aspergillus flavus and Aspergillus parasiticus. They can grow in corn, cotton seeds, peanuts, walnuts, almonds, soya, rice, and who knows what other grains. Now, if we look at their structures, these are polycyclic furan compounds that possess an intense blue or green fluorescence under UV light. There are four major aflatoxins. If they fluoresce blue, they are B1 or B2, and if it is green, they are either G1 or G2. An aflatoxin M1 is the one found in animal products like milk. Most problems occur as a result of drought and insect damage, which allow the mold to get started, followed by a prolonged hot weather. They also like high relative humidity in the air, above 85%, or a corn moisture content above 14%. So remember, temperature and humidity must be there for proper growth of most funguses. The sensitivity in animals varies a lot according to the age and the species, as shown here. From the most sensitive to the least sensitive among domestic animals, we have ducklings, trout, dogs, horses, swine, bovines, and chickens are the most resistant. In general, most animals are generally more sensitive. Young animals are more sensitive than adults. For example, doses that can be acutely lethal to piglets of between 400 and 600 parts per billion are okay in pregnant sows and boars, and they do not, and you wouldn't expect to see any reproductive problems. In other words, there is no effect on spermatogenesis, litter size, piglet survival, or lactation. So what are the clinical signs in piglets? Well, not very specific, as you may expect. Poor weight gains, anorexia, poor body condition or unthriftiness, rough and dull hair coat, and also an increase in prevalence of other infectious diseases, because these are immunosuppressive toxins. If the concentration in the diet is more than 500 parts per billion, they will show the same signs as before, but on top of that, they will also have hemorrhages, petechia, melena, fluid accumulation in the abdomen, ascites, and all of these signs are attributed to liver failure. Now, we're going to talk about the levels of toxicity in a minute, but the mechanism of action is through binding to proteins and DNA. It is a very potent carcinogen in both animals and humans, and is not only known for producing liver damage, but also carcinomas in humans. The toxicity on the immune system is also important because it makes the animal prone to infectious diseases. There are numerous studies that look in detail at the mechanism of action. I got this image from a review paper that was published in 2020. It explains well how the genotoxicity takes place by interfering with gene expression, DNA integrity. It inhibits repair mechanisms, and this explains why it's so carcinogenic. It also explains how the tissue necrosis is produced, as well as the interference with both the innate and the acquired immunity. Now let's look at tolerable levels for humans and animals. You do not need to memorize any of these, but it's good to know that FDA issued some action levels for humans. All products except for milk should have no more than 20 parts per billion. As you can see here, for milk the action level is just 0.5 parts per billion. And this table is only for corn, actually. So I included some recommendations at the bottom that are more relevant for pigs and cattle. Less than 50 parts per billion are safe for any animal. If it is higher than 50 parts per billion, it should not be fed to weanling piglets. If higher than 100 parts per billion, should not be given to grower pigs. and less than 300 parts per billion are acceptable for finisher pigs. For calves less than 4 months of age, the diet should have no more than 50 parts per billion. But if it is an adult cow or bull, they can tolerate up to 500 parts per billion. So what are some options available when the food is contaminated with aflatoxins? Well, one of them is using Novacil, which is a binder approved by FDA for anti-caking. And there is research that at concentrations of up to 3,000 parts per billion of aflatoxin B1, 0.1% of Novacil was effective protecting pigs and chickens from all the toxicity actions. They grew at similar rates to the controls, and the animals had no lesions whatsoever. Ammoniation is another option because it breaks down the aflatoxin structure. The downside is that it turns corn brown, but if it is going to be used for animal consumption, it's a perfectly viable option. And as we saw, there is big difference in toxicity between species and ages, so we can use contaminated feed to less susceptible species or ages, or maybe dilute it with clean feed if you have that option. And finally, we're going to describe a feedlot case that was published by folks at the Iowa State University back in 1985. There were approximately 200 steers out of 14,000 animals weighing between 200 and 300 kilograms that were ill on December 23rd or by December 23rd, with 40 already dead and a total of 100 animals dead by January 3rd. The clinical signs were of lethargy, ataxia, weakness, and upon necropsy, the grossly visible lesions were swollen and firm livers. There was edema of the gallbladder and the mesenteries. There were large accumulations of fluid in the thoracic and abdominal cavity. And when they did histopath, they saw hyperplasia or proliferation of bile ducts, diffuse vacuolar degeneration, and fibrosis. They reported that 1 month earlier, there were quite a few animals with bovine respiratory disease complex, but when they did necropsy, they did not have any liver lesions. So only those that were necropsy after December 27 had liver lesions. They were feeding about 35% of whole cottonseed, 35% corn, 19% gene trash, 10% molasses. And when they did the chemical analysis, it revealed very high concentrations of aflatoxin B1 in the cottonseed and the gene trash. They collected at least 10 samples from each ingredient, because there can be areas that could be hot spots, and there could be other samples with very little or no aflatoxins. So this makes the interpretation of the analysis in feed very difficult, as you can imagine, because the toxic dose ingested can never really be defined or reached in field cases. They also mentioned that 50% of the livers among cattle from affected pens were condemned because of the jaundice and fibrosis. What they did to confirm the exposure was detecting aflatoxin M1 in the urine of the 5 animals. So this is the final slide. On this next video, we will continue talking about more toxicants that affect the liver. So take care for now, and I'll see you on the next one. Bye-bye.